and it's my pleasure to be here presenting some work from our Brain Aging Network for Cognitive Health, or our Healthy Aging program. And the first thing I want to just mention is this is our team. We span faculty, postdoctoral fellows, and really most importantly, research coordinators. They're the backbone of our program. These are the folks who you interact with most often when you come for your visits. And I just want to acknowledge the incredibly hard, diligent work that they do to make this whole center run. This is also a bit personal for me because I was a research coordinator for our Healthy Aging program. I recognize some familiar faces from my time here, and it's really just an honor to be back and share some of the science that we've been doing recently. So this is really what we focus on in our Healthy Aging program. There's a few observations that we have with aging. We know that to at least some degree, all brains age, but we also know that some brains age slower than others. And this is really important because we want to understand why do some brains age more slowly than others. What I've highlighted here on this figure is the y-axis is a measure of cognitive performance, and the x-axis is chronological age. And you can appreciate that with advancing age, cognitive performance declines. But highlighted there in the red rectangle are some really phenomenal cognitive agers. And we want to unlock what is it about these excellent agers that allows them to maintain high levels of cognitive function with advancing age. We're going to talk about a few of those today, and Gil had alluded to some of those factors, and I'll hope to go in a little bit more detail. And so the ways that we think about what questions we want to ask is, what is the biology driving cognitive aging and brain aging? If we can understand the biology, we can maybe develop more targeted treatments and better biomarkers to identify folks who are at risk for slower aging or faster aging. And then how can we modify this with our behavior? So our program is really a data collection gauntlet, as many of you know. And we think about a number of risk factors for cognitive aging. Some of these are non-modifiable, like chronological age or biological sex or your genetics. Maybe some of them are semi-modifiable. And then we have a lot of modifiable risk factors like exercise and diet and sleep. And when you come in for your research visit, you may do a number of things that help us better understand what these risk factors are doing to the brain. When we think about biomarkers, we do brain imaging. So we look at the volumes of your brain and the white matter health in your brain. We look at PET imaging to measure amyloid plaques and tau tangles. And we measure your biofluids. So we'll collect blood, spinal fluid for those who volunteer. And this is a big area of focus for my research, particularly where we can measure some of the proteins that cause Alzheimer's disease, as Gil mentioned, as well as many, many more proteins in blood and spinal fluid that allow us to measure other biological pathways that we think are very important for cognitive function. So when it comes to cognitive function and behavior, we run you through the gauntlet of our memory tests. We do questionnaires to better understand how you're functioning in your daily life, mood, any symptoms that you might be experiencing. And we also do a lot of remote data collection through wearable devices like a Fitbit. This allows us to measure your physical activity, sleep, heart rate, and get a more comprehensive picture of your day-to-day behavioral health. So first thing I'm going to focus on here are some of these cornerstones of cognitive aging, and particularly chronological age and biological sex. So when you come into a research visit, we give you some of those awful memory tests that a lot of people don't really enjoy. But when we do get the data, we can see some very clear patterns that are very reproducible. So this is actually from the Baltimore longitudinal study of aging of over 2,000 older adults. What you can appreciate is that with older age, memory declines, and we also observe a long-standing sex difference in that women tend to outperform men on our tests of memory. We find this really fascinating because this is something that we observe across the lifespan. So it's good news for maybe half of the folks in the room. But what we'd like to do is understand why, so maybe we can help close the gap for the fellows in the room. So one way that we've approached this is using the really rich information about the brain that is present in spinal fluid. So we conducted a study with folks from our program, 95 of you, typically aging adults, roughly 50-50 in terms of male and female. And we did what's called proteomics. So we looked at spinal fluid that was collected and measured over 7,000 different molecules in spinal fluid. Our goal was to then screen the spinal fluid to identify what are the individual molecules that are most strongly associated with age and sex and episodic memory. So like I mentioned, we do the memory testing, and this is really what we found. So this figure on the x-axis represents the relationship between every individual protein and chronological age. Anything in red is a protein that increased with older age. Anything in blue is a protein that decreased with older age. Those proteins were also color-coded based on the relationship with memory. So as you might expect, proteins associated with older age also were related to worse memory performance and vice versa. We did the same thing with biological sex, and we saw similar patterns. So proteins associated with female sex in red were associated with better memory. Proteins associated with male sex in blue were associated with worse memory. We then took these lists of proteins and we annotated what are the biological pathways or molecular roles for these individual proteins. And what we found is that proteins associated with worse memory that were also related to age and female sex were linked to higher levels of brain inflammation. So we think that inflammation is a really important component of memory aging. When we looked at proteins that were associated with better memory, we identified markers that were really important for our synaptic connections, how tightly linked different brain cells can communicate with one another. So these are additional proteins and pathways that we are very interested in developing biomarkers for. They may help us in the clinic for screening who's at greatest risk of cognitive decline. I don't need to go over this figure since Gil introduced it. We're very interested in all of these individual factors that lead to potentially 45% reduction in dementia cases. Today I want to highlight sleep and a few other factors. So this is work led by Emily Paolillo, an assistant professor in our program, and also Salma, who's a research coordinator who you might have met today, has also done some of this work. So what Emily has found is using the Fitbit devices that you wear during sleep, we can measure how long you're sleeping during the night. And if we just look on the x-axis, average duration of sleep through the night, we see that there's a negative association with the measurement of the Alzheimer's disease biomarker in your blood. So this is really important because there's epidemiological evidence that sleep is a risk factor for Alzheimer's disease. And here we can measure, with you sleeping at home, with the blood test, that having more optimal levels of sleep is associated with less Alzheimer's disease biology in your blood. But how long you sleep is just one facet of sleep. There are so many other measurements of sleep that we can acquire by monitoring through the Fitbit. One interesting pattern that we've looked at is the consistency in the timing of sleep. So the graph on the left, that large column with the thick blocks, represents every night of sleep over the course of a 30-day period. And what you'll appreciate is that this individual went to bed at basically the same time every night and slept roughly the same amount of time every night. When you look to the right, you can see a very different story. This individual sometimes went to bed at 9, sometimes at 2, sometimes they slept for 3 hours, sometimes they slept for 10 hours. It was a very fragmented and irregular sleep pattern. When we develop a measure that basically captures the consistency and the timing of your sleep, we have also identified some very interesting patterns. What we can see is that those with more regular sleep had larger brain volumes. And so we think that this is a very important measure because we controlled for how long you slept in this analysis, and it was still important in predicting your brain health. So we're starting to learn a little bit more and get a little bit more precise so we can get better recommendations as to sleep hygiene. Physical activity is a big one. We all know that physical activity is good for you. We've known this for a very long time. We've known this since we were very young. We're very interested in what aspect of physical activity is most important for brain health and also why. So Claire Cadwallader, a postdoc in our group, has really taken the task of synthesizing all of the physical activity data that we get from the Fitbit devices. And this figure on the left is an example of some of the information that you can gather from the Fitbit. She used step cadence, which is a measure of how many steps you take per minute, to identify epochs or periods of physical activity, which she defines as sessions, highlighted in the green there. Using a measure of the frequency of the number of sessions of physical activity that you engage in, we can then look at that in relation to measures of brain health. What she observed was that even just engaging in some physical activity sessions was related to better executive function, or another measure of judgment and decision making that we have on our cognitive testing. And some key takeaways from her work is that really any activity is better than no activity. And shorter sessions at higher intensity and frequency may actually be optimal. So you don't need to work out necessarily for 1 hour, 2 hours, 3 hours. Even a little bit of effort at a moderate to high intensity may be important for improving your brain health. There's obviously a lot more work that we need to do. We know that there's different types of physical activity. It's not just steps. There's swimming, there's weightlifting, and we're going to work to better understand those different dimensions. So going back to this idea that we can measure thousands of proteins in your blood and spinal fluid, we wanted to now ask, okay, what is the biology driving this relationship between exercise and brain health? So again, we turned to proteomics. Using 65 older adults who had the Fitbit measurement and plasma proteomics, we asked what are the individual markers that are most strongly associated with physical activity. So the figure on the left, the x-axis represents an individual protein's association with step count over the 30-day period. The y-axis is a measure of how strong that statistical association was. And we can identify hundreds of proteins in blood that are related to physical activity. What we noticed when we looked at the biology represented by these are proteins that have previously been linked to aging. So this is giving us evidence that physical activity in later life can slow down molecular markers of aging or more youthful protein levels in your blood. And importantly, we saw that this was related to better cognition. So our top marker, ANTXR2, which is a marker of vascular plasticity, or how quickly your blood cells can recover from injury, those with higher levels at baseline showed less steep cognitive decline over time. So we're very interested in modeling the biology of exercise so that we can better develop treatments that recapture these observations that we've known for such a long time that physical activity is good for your brain. Finally, I want to touch on some very recent work from our group that's looking beyond person-specific factors, but really thinking about the environment and space that we live in. So we want to understand, how does ambient pollution affect the aging brain? Work from Gill's lab has shown that higher levels of Alzheimer's disease biology has been associated with higher pollution in the neighborhood or environment that you live in. We know that there are other neurodegenerative diseases that may be at risk due to ambient air pollution. We know that this is a really important social determinant of health. Where you live in your neighborhood and the socioeconomic factors in that area are directly linked to pollution in the environment. And so we wanted to better understand this phenomena in the Bay Area. So what Sophia, a research coordinator in our group, started to look at is she looked at a pollution map of the Bay Area. So this is an example of a pollution map in San Francisco. By and large, San Francisco is doing pretty well. You can see a lot of neighborhoods in green, but there are neighborhoods in yellow and red that are experiencing higher levels of pollution. And what we were able to do is link pollution levels in the neighborhood to individual cognitive performance. And what we observed when we were looking actually here at a measure of white matter health in the brain is that higher pollution burden score was related to greater white matter hyperintensities or markers of white matter injury in the brain. Now, I will note this is a very small effect, but it was statistically significant. You could imagine that if you were in an area not like the Bay Area, but an even more polluted area, that this effect might be even larger. So this is early work for us, but we're very interested in understanding what are the pollution factors that might drive brain health, and this might be very important for us as we think about policies to try to improve the environmental conditions that we live in. So with that, I just want to thank you all, again, reiterate, participants for really contributing your time, your effort. It's a heroic effort, and we're really so grateful. Thank you to our funders and our AG team.